So welcome everyone to the Connect Science seminar series organized by the FHCC. Before we start, please, I just want to let everyone know that you should write your questions in the Q&A option at Zoom, and I will read them to Dr. Krummel at the end of his presentation. And the seminar will be posted in the website afterwards. So if anyone misses it, which would be really bad, you should not miss this presentation today. You can see it there. And this is great to have this opportunity. Actually, I was thinking that while it's snowing here in Boston, we can have Dr. Krummel presenting to us from probably nice weather San Francisco, or not too cold weather San Francisco. So my name is Judith Agudo. I'm an assistant professor in the Cancer Immunology and Virology department at the Dana Farber Cancer Institute. And it's today really a true honor to introduce to all of you Dr. Matthew Krummel. I don't think he really needs introduction to anyone that works in cancer immunotherapy, but just for people that work in, let's say, other topics related to cancer. Dr. Krummel is a professor of pathology and the Robert Smith Endowed Chair of Experimental Pathology at University of California at San Francisco. There he's also the chair of ImmunoX program. I see he has the t-shirt, so that's an outstanding program for training of immunology trainees. Dr. Krummel has a very long history in cancer immunotherapy, starting already during his PhD with Nobel laureate Jim Allison and early work in CTLA-4, then postdoctoral work with Mark Davis in Stanford. And he has a really rich plethora of papers related to autoimmunity and cancer. And in the last, I would say, decade, I think he has done something remarkable, which is that in my opinion, a little bit very centric T-cell checkpoint blockade field, he has taught us the importance of other cells, in this case, dendritic cells in the response, the immune response against tumors. So first in the primary tumor, then in the draining lymph node, how they transfer antigen one to another. In a metastatic setting, how early disseminated cells can get, let's say, surveyed by these dendritic cells, and in particular cross-presenting dendritic cells. And once he taught us all about cross-presenting dendritic cells, he went and showed that actually DC2s, which are the other dendritic cell, the ones that present to CD4 are also very important in the response to immunotherapy. So anyway, it's really a pleasure to have him present here today when we've seen, we've had his paper so many times for Journal Club in the lab. So having him presenting the work in live, it's really an honor. And without further ado, please, Dr. Krummel, thank you so much for accepting our invitation, and I look really forward to watching your presentation. Thank you.
Well, thank you so much, Judith, for that really kind introduction, and thank you all for the invitation to come today and for being here. So let's go. So I want to talk today about this concept that we've been really elaborating over the last, as Judith said, last sort of 10 years. And I'm going to kind of give it the historical introduction that we've been at this problem of trying to figure out how the immune system works in general. And at one end of the spectrum, that involves the question of how, for example, molecules work on the surface of an immune cell. So how does the process of recognition, where a T-cell receptor affinity is able to bind a peptide MHC complex, how does that function? How does that actually work? And one of the things that you learn as you start to think about that is that the way that the T-cell receptor or cytoskeletal molecules assemble are ultimately dictate to a great extent the way that the subcellular interaction of an immune cell works. So the entire T-cell surface is a composite, obviously, of things like T-cell receptors and membrane and underlying cytoskeleton signaling molecules. And so that's an emergent behavior. That's in sort of those terms, the subcellular definition of an immune cell is a function of all these interactions that are very definable at the molecular level. So I want to pause there for a moment, because that's one of the things that we use as a jumping off point a lot in our lab is is to look at subcellular behaviors, to image it, and to start to use that as an entry point to launch hypotheses about what's going on. So I want to use a lattice light sheet image of this particular interaction as a point of reference for today's talk. So T cells here are in green, and you can see their little microvilli, and in red are a dendritic cell. Blow that up here a little bit. And as this movie plays, I want you to get the sense of how dynamic and how really convoluted that interaction is. So T cells are busy using their microvilli to palpate the surfaces of the things that they want to see. And on the other side of this, one of the things that becomes really apparent when you take this apart is how much work the dendritic cell does to be seen. And so at the same time that you have this really the convolution of the T cells that are doing these little probing behaviors that you see here, the dendritic cell is very much conforming to that surface. It's putting its membranes sheets out, and although we call them dendrites, they're actually more like veils that form that junction. So I like this movie because in the context of today, particularly, it really shows this idea that immune systems are partnerships, that as much as the T cell wants to probe for peptide MHC, the component parts, the immune system that work with it, really work with it as opposed to being passive, things that get observed. Oh, so if you build from there, the next sort of level of complexity that we get up to is the idea of multicellular. So if this is a single cell interaction and we have these partnerships, if you take something like a tumor microenvironment, so the black here is where the tumor is, and these are various different probes that label cell types. This level of complexity obviously is a function of what the cell types are and what they're trying to do at this subcellular level. So the multicellular level is an emergent behavior of all the different cells that are present below. And of course, the way that we really want this to have meaning is at the level of health. And so there's a final emergent behavior from all these multicellular interactions that is really the one that determines, for example, in cancer treatment, whether people die or whether they live. So that's really the ultimate emergent behavior in some ways of all these interactions and all their different levels. So today I want to focus on this side of what the lab works on and get into this area that we've been sort of trying to elaborate this, we refer to as archetypes. And that is the collection of cells and their individual identities that give rise to a tissue and its immune state, and ultimately then define this aspect of health. So Judith gave a nice introduction to the work that we've been doing, and started with CTLA4, and it's been elaborated out into some of these other cell types that I'm going to talk about today, the archetypes. But I find it necessary to kind of reflect back that while cancer immunology has had these huge leaps and bounds in the last 20 years or so, outside immuno-oncology, the immunologists have been doing a lot as well. And I don't mean to be patronizing to this. I want just to emphasize this because of how important it is to cancer immunology, and the cross-fertilization between those fields. So just to remind you, and this, for some of this will be evident, and in the last 10 or 15 years alone, the role of the immune system in the brain and everyday behavior is become hugely evident. And this is not your parents' immune system where it's just about defense. I guess the work maybe the most famous is maybe in the complement field, that if you eliminate just complement proteins and obviously the microglia that are associated with them, you will not remember today's talk. And that's because short-term memory is a function of the pruning behavior of microglia, which are the macrophages of the brain, on neurons. And so your immune system in that setting is doing this constant pruning behavior of a tissue to make it sort of the homeostatic best that it can be, let's say. So that again is not the sort of the immune system you think of that is either tolerant or destructive, like you would want to be against a major pathogen. It's in this inner zone. Similarly, in obviously the microbiome is an example of that same theme. That there, rather than your immune system attacking everything in your gut, it's not ignorant to it. Your Tregs and some even T cell subsets are acutely tuned. They are essentially curating the bacteria of your gut. They recognize it, but rather than mounting a huge immune response, they may make very subtle cytokine changes that allow bacteria that's making vitamin K to be present there. So that's definitely an example where the immune system, examples where the immune system is doing a lot more than just those two ends of the spectrum that we've learned in basic immunology. Other ones, for example, arteriosclerosis, when at least I was being raised that everything was about diet for arteriosclerosis, the presence of lipids in your blood. We now really understand that underneath our arteriosclerotic plaques, there are angry macrophages. And so all these immune interactions and all these different tissues, and we could go through each one individually, but I think you probably know them all, really represent the spectrum of the immune system. And that really brings up the idea that the immune system has not just at one thing, the ignorance of pathogen versus responses to pathogen, but has all these possible states that it can get into. And some of those states are really nurturing, like in the brain. In the case of Alzheimer's, perhaps those you get into the wrong state, and that is somehow self-reinforcing to the point that that's chronic disease. So, to me, understanding tumors that don't respond, for example, to existing immunotherapies, likely starts with understanding these goals. So if we take the spectrum of the immune system, we just kind of conceptualize it. Again, in the past, we would have thought of tolerance at one end, where the immune system doesn't recognize things at all and is sort of relatively inactive to them, to the concept of something that is very reactive. You've been vaccinated against something, and you have a host of T-cell receptors that are floating around, and antibodies as well. This whole interzone is way richer than I think that we realized 20 years ago, and we start to understand them. So if you start to take apart something like commensal symbiosis, that role for the immune system to recognize and curate commensals, we start to identify cell types that are present within that sort of archetypal collection of cells that are important for that to take place. But having said that, any given member of that can be found in other settings. So for example, Tregs are really also important in tissue metabolism. So we now understand that Tregs, together with macrophages, are really important in regulating fat metabolism. And dendritic cells that may be involved in commensal symbiosis can also be involved in other settings as well. So the idea then that we've been trying to launch is an order of abstraction above the cell type that represents the collections of cell types. And that's what we're sort of here calling out archetype. It's a collection of cells that are in linked states across tissue types, and probably following an evolutionary design where the immune system is meant to be nurturing, or in other ways helping homeostasis. And I think by understanding that, we're going to start to get in a place where we can see tumors as collections of these. These are relatively stable immune states that are designed for something else, and all a tumor needs to do is dial in one. And some tumors may dial in wound healing responses, and other ones may dial in responses that are meant for tissue metabolism or combinations thereof. And so really understanding these becomes a critical part of seeing cancer immunotherapies work. Okay, so just to briefly put that in the perspective of another thing that we'll sort of jump into, is that if we call this sort of area of the immune system accommodation, this is generally putting all these states and these behaviors into a class. At the other end is the stuff that we care about, and that also exists in classes. So I'll start out there with the question of, what are the sort of archetypal immune collections that are necessary to get destruction of a tumor? Okay, so for us, this started with imaging, when we started to observe that T cells in tumors seem to stop and have partnerships, have the same sort of synaptic interactions that I showed you in the movie. And this is where we really discovered the key role of CDC1s that Judith alluded to. And just to give a little historical context to that, even prior to the work we had done, there was evidence that CDC1s might be involved in the fact that Ken Murphy had knocked out the critical transcriptional factor BATF3 and seen in the EL4 model that there was impaired anti-tumor immunity. At the time, they attributed that to CD8 alpha dendritic cells, which are a subset that are also BATF3 dependent. But by the time we sort of came along in 2014, there was this real question of, are there any allies in the tumor microenvironment on which a T cell response is going to be made? And if so, what are they? And so Miranda Burrows, who was in the lab at the time, put together what I think is a pretty seminal paper that showed that if you purify all the different antigen presenting cells from a tumor microenvironment in a setting where there is a model antigen, ovalbumin, and you mix them with T cells, and you see which ones can induce proliferation. Sort of not unsurprisingly, the tumor-associated macrophages don't upregulate NER77, don't lead to upregulation of CD69, don't lead to proliferation. Although I'll say that in an emerging story, the tumor-associated macrophages very much form synapses with T cells. But it was really only the CD103 dendritic cells that were able to induce that, and that's the green shifts you see in these plots. At the time, she also showed those are the major cells that produce IL-12. They're also the major cell types that do cross-presentation and can keep antigens in a relatively neutral environment. And in fact, if you look at overall survival now on here on the right versus time, looking at just the gene expression signatures of these cells in human patients, you can see that there's a really nice correspondence between people that have high sort of evidence of these cell types being present and survival versus those that don't. So this has really been a cottage industry, and a number of groups have contributed and continue to elaborate this story. Lisa Cousins and Brian Raffel, to name a couple, Mary Murad, local to the East Coast, David Sancho have all used BATF3s to show that this is what's required for PD-L1 responses in mice. Tom Gajewski and Kitano Souza looking at CDC1s in mouse models and how they might be elaborated. And really, this is starting to be a question of what is the underlying reason why CDC1s exist in these environments. And so we started to address this question by a huge program that's been very fruitful at UCSF called Immunoprofiler. And this is something that we set up as a way to provide sort of a conduit for all comers tumor samples that any clinician might want to bring to us to dissociate those and do high dimensional flow cytometry to profile what's in the tumor, and also to do various forms of transcriptomics to look what's in component, either collections of T cells, or now 5 years later, everything is obviously single cell sequencing. And so one of the things that we were able to do with the data set that we had already collected from that related to the CDC1s was to ask the question of, if you take total RNA from a tumor, and you already know which tumors have large numbers of the CDC1s, what genes expressed in the tumors are indicative or correlated with that? And one of the really interesting ones was FLT3 ligand. And so this is interesting because FLT3 ligand is sort of a pseudo cytokine. It's a cell surface associated cytokine, so maybe not even a cytokine at all. That the more you have of it in a tumor, the more you had CDC1s. So it kind of makes sense, although you wouldn't have necessarily thought the source was in the tumor itself. So somewhere in the tumor, there is a cell that is making FLT3 ligand, and we wanted to know what it was. And so we made a FLT3 ligand reporter in mice. And this is just like the design you'll see all around. If you look at the top here, the FLT3 locus drives teal fluorescent protein. And when you look into the tumor, you really don't find some of the cell types that people had anticipated to express this, maybe stromal cells. But in fact, you find that NK cells and CD4s and CD8s lymphocytes are the predominant producer of this cytokine that makes CDC1s. And that's just quantified down here at the bottom. So that's the flow cytometry quantified here. So cutting that story sort of very quickly, we went to, again, the Immunoprofiler data set. And we took melanoma patients and we parsed them by whether they had been responders to PD-1. So patients are in columns here and cell types are in rows. And so we parsed them and we said, what are the cell types whose presence has the highest p-value association with being a responder versus a non-responder? And by that virtue, the cell types that have the biggest ones are the CDC1s. Sorry, for those that don't know, CDC1s express PDCA3. So these are those dendritic cells. So you can see that responders are red hot for those in general, non-responders blue. But the co-associated cell type was in fact NK cells. And that's, again, in the mouse models can sort of corroborate that NK cells are one of the key sources for the cytokines that make CDC1s locally. And that really brings up this question that we don't have the answer for just today as to what is it that brings the NK cells there? So this starts to be that explosion of the archetype to say, okay, well, what is it about that is being mimicked that makes for a responsive immune system, a responsive archetype? So this is what we started to call type 1 responders, and you can see them all the way over here to the left. But a careful eye will notice that there are some patients that were responders, even in this small sort of 15 or 20 patient data set, that didn't have that feature that I've just defined for you. And if you look down below at the other cell types that are really associated, then this is a hierarchical clustering of the responders, so that each one cell, each person that is put next to their most similar. You can see that the next sort of cell type that emerges are CD4 cells down here. So this phenotype suggests that there's a kind of a second set of people that respond to checkpoint blockade. So if I plot for you in the responders over here, BDCA3 positive dendritic cells is black. You can see that, for example, the non-responders, there's very few BDCA3s. So this is the association we're referring to. You need to have these dendritic cells in order to make a good response. But you can see kind of a waterfall of expression amongst the responders. So you have all these people that are generally high. Everybody's higher than the negatives. But in those that drop off, you start to see the people that don't have as many CDC1s actually start to have BDCA1 CDC2s. So they have an alternate dendritic cell subset that basically correlates with the CD4 response. So we start to call these type 1 responsive archetypes. So that is in allusion to class 1 CDC1s, and which are here. So that's these people. And then type 2, allusion to class 2 and CD4, are these people that have in general CDC2s instead. So another way of looking at that is to just define what we've learned about this. And I'm going to cartoon this for today in the interest of time and to kind of get through some material. So in Kevin Berry's Nature Medicine paper of 2018, we showed that part of the archetype that makes a type 1 reactive immune system involves natural killer cells that deliver FLT3 ligand to help generate CDC1s. Through a series of papers, ours and others, we know that CDC1s have at least one of their functions is to migrate to lymph nodes and to present to new CD8s. But a second function of the CDC1s is to help those CD8s by re-stimulating them in the tumor microenvironment so that you end up with robust rejection. So this starts to look like the beginning of a reactive archetype. And it contains 3 major cell types. In this case, a source of FLT3 ligand, which is typically NK cells, the CDC1s and CD8s. And we could go in questions and deeper into what defines the CD8s that one finds there, or what are the unique features about this. And many labs, including Mikhail Pitets in your neighborhood, has defined some of the features that make CDC1s particularly good. In fact, some of the differentiation comes into question, like, are CDC1s always CDC1s? So on the other hand of the spectrum are these other type of responders to checkpoint blockade and sort of with responsive archetypes. And these involve CDC2s now, as I said, that ultimately have a similar feature. They go sort of chronically, they'll go to the lymph node and present antigen. But if they are not regulated by regulatory T cells, they will stimulate those like gangbusters. You end up with a really nice phenotype of ICOS high, PD1 low CD4s being generated, and those can independently reject tumors. So this is a kind of a second reactive archetype, and I think we're starting to understand the hooks that go into these. So the reason I bring this up is because it starts to allow us to take apart the world of patients. And again, that's where you go from the multicellular into the clinical. So if I were to diagram these sort of reactive immune archetypes that I just defined, we have those people who look like, I call this an antiviral response sometimes, a class 1 response of CD8 T cells, CDC1s, and NK cells. Looks like probably something that was built for antiviral responses. You also have something that may be the antiviral class 2 or maybe against intracellular bacteria, CD4 T cells, these CDC2s. And if you look at these patients overall and you find these evidence of these cells, they're basically already a decent overall survival. And then you start to get into patients that don't have those because one way or another, maybe they have a large number of regulatory T cells that turns off this, or features that we don't really understand that I'll get into in a moment, that essentially you end up with poor responses to checkpoint, but also worse overall survival. And so we start to be able to categorize patient immune systems by these things. And what's neat about that is that you can start to put them into disease classes. So for example, melanoma, the indication about 50% of people respond to checkpoint blockade, and this is sort of their breakdown. The 50% is roughly 35 or 40% of them look like they're CD8 responders, and the rest are CD4 responders, and then 50% of the people don't respond because of immune systems that I'll try to get into a little bit in the next little section. Whereas head and neck cancer, we hardly ever see evidence of these class 1 immune systems. And so as we're starting to see the clinical trials coming out from this, anticipation is around 30 to 30% of the people will respond because they have this CD4 immunity that doesn't have regulatory T cells bearing down on the CDC2s. Okay, so I'm going to take a little foray before moving into the COVID section of the talk, which will also be about archetypes, to get into the question of how we're going to go forward with this. And this is a still taken from a movie that I won't play for you, but I will just describe for you that when we watch CD8 T cells in this is a breast cancer model in mouse. When we watch them, we find that the CD8 T cells largely cluster around tumor-associated macrophages here, green, yellow, and orange. So we'll see these swarms of T cells in these regions, and you can see other ones here. But then very occasionally, we'll actually see the reactive archetype that I described. Namely, we'll also see CD8 T cells swarming around red dendritic cells. And they actually turn a different hue of blue, and that's because their pH changes, and CFP is pH sensitive, so we can actually see that in the microscope. But it's super rare. While this biology that I've just been telling you about is ongoing, this reactive CDC1 and K, for example, the most of the immune system is taken up doing these other things. And I think this is where my introduction of reminding you about the immune system in the brain and the metabolism and commensals and lung fibrosis comes into play, that there are these other roles for the immune system. And these are the dominant archetypes. This is the dominant biology that's going on as the tumor dials in the immune system for its own purposes. So we've been looking to uncover those dominant archetypes, both with regard to the garden variety of tumors and in regard to space. So I'll tell you about the garden variety first. So this again involves the immunoprofiler initiative, and this is a cartoon of a study now where we have about 300 different tumors from a whole bunch of different tumor types. And these just represent in parentheses the numbers of cells of patients that we've used to create these density maps. So again, the idea is that every patient gives us a biopsy, a tumor biopsy, usually as part of a resection, but sometimes separately. And from that, we can end up getting scores for various different flow populations, the frequency of CD4 T cells, the frequency of regulatory T cells. And we can also get gene expression profiles that either can be used to go back and forth between this or actually to find expression profiles. And so doing this, you can, for example, you can take patients across different indications and just say, what is their composition of the immune system just looking at T cells? And you find that hepatocellular and head and neck and kidney are very rich in T cells, whereas GBM and PDAC are fairly low in T cells. You can do the same thing for myeloid cells and stromal cells, and you find, again, you can waterfall it, although the order is not the same. So for example, kidney has both high numbers of T cells and myeloid cells, whereas something like head and neck has a large number of T cells, but actually not so many myeloid cells. So that's just, that at some point helps us to understand the garden varieties of tumors compared to each other with respect to whether the immune system is generally present or not. But the other thing it really points out, and I'll point to it really obviously here in PDAC, is that it also points out to the heterogeneity of immune systems within tumors. So for example, in PDAC, you can really see that although we plotted this as a box and whiskers plot, which is what you should do if things are mean-censored, you actually can see that this looks very bimodal. And most of these, in the end, look like there's quite a spectrum of distribution of immune system within them, such that some patients are very high and some very low for any given feature. So this suggests the idea of a clustering algorithm. And so we can do this clustering algorithm simply as a frequency. Say, okay, if we just ask about frequency of immune cell populations, what sorts of things do we find? And so if you even just take T cells, myeloid, and stroma alone, you start to create, you can see, you might have thought that would be for high-low, that there might be 8 combinations of high-low, high-low. In effect, we only ever see 6 that cluster out together. And And those create sort of pictures of, or sort of, again, archetypes of patients that are immune rich, that have high T cells and myeloid populations, but maybe not so high in stromal populations. And so this starts to be a way that we can take patients across indications and ask about their composition. So I'm going to fast track through this to the point that you can go to many, and you can start to include more and more features, and sort of you can use an alluvial plot to see how your numbers of archetypes, your collections, start to somewhat get bigger over time, so that you end up with larger and larger numbers. Although really we get to about 12, and then they don't branch very much anymore. And if you do that and you say, okay, so for example, some tumors are CD8 biased, and they're also really macrophage rich. They don't have as many dendritic cells. That represents a dominant class of the types of tumors we see. That tends to have a lot of kidney cancer in it, for example. You can classify tumors based on their composition in this way, and then you can start to ask, is that meaningful? Does that predict anything else about the tumor? And I won't go into huge detail in this, except three plots that I'll show you. So one of them is if we look at the RNA from the live compartment and we start to apply signatures for cell types that we didn't include in our clustering. So if we clustered based on myeloid composition, T cell composition, stromal composition, and we come back and we look for an NK genes, we can find that certain archetypal collections are where all the NK cells are. And that's not surprisingly, it's also where the CD8s are. Plasma cells and B cells also tend to show up with particular collections of tumor microenvironments. So this starts to show that what you can do is, if you start to look by composition alone, that you can start to find classifications that predict other parts of the composition, even the ones that you didn't include in your clustering algorithm. And that extends to, for example, chemokines, that if we look and we arrange these 12 archetypes that we have at this point that represent different compositions of T cells, et cetera, that the chemokines start to cluster by that too. So if we look at chemokine expression within the tumor microenvironment, we'll find that there are dominant chemokines, and many of these are very nicely explanatory. So some of these are T cell chemokines that correspond to these very T cell rich archetypes. And then the last part of this that I just want to point out is that the tumor biology itself likely has something to do with this. If we start to organize genes that are, for example, interferon stimulatory genes, senescence EMT, we see this distribution of spattering, so that these sorts of archetypes of immune systems tend to have a correspondence to ISG rich, whereas these sort of more deserty tumor microenvironments tend to be ISG low. And conversely, you can find fibrosis associated genes, for example, associated with one or more of these deserts, but not so much with the immune rich regions. So this is an area that we're really actively pursuing to try to find out what sorts of immune systems are actually being co-opted in cancer, to obviously understand what sorts of things you'll do about different ones. So the other side of that is that we've been trying to get into this question of, if you're in an archetypal space and you're starting to understand what's going on, how do you find the rest of the connections? So I mentioned the NK story, and that came about sort of organically by finding FLT3 ligand was associated with CDC1s. We can now get into this world where we can take apart a tumor and start to cluster its cell types by single cell sequencing. And we can find out all the heterogeneous cell types that are present. And separately, we can image these, but we have a really hard time finding the connections between these cell types if it's not simply by composition, like I just told you about. So how do we, one of the more likely things is that physical space, or what Gary Nolan calls neighborhoods, will be the place where we'll find some of these interactions that we want to know about, but we need to be able to find that cell type. And so this really gives you very limited information about the spatial. And so I'm going to tell you about a little technology that we've been working on for spatial sequencing following imaging. And it looks like this. So the idea is you want to end up with your single cell sequencing cells, and you want to know who was whose neighbor. And so you want to know who was in the same neighborhood with one another. And so in order to do this, at the end of an imaging run, we can perfuse in an antibody or a lipid that contains a barcode. That barcode is a heteroduplex that has an overhang, but the overhang is blocked. And the reason why it's blocked is that ultimately we want to print to it, but we want to print to it in a light dependent manner. So the blocking, the caging moiety is a photo cage. And ultimately then the idea is that we're going to be able to add a strand to here that has some sort of read that allows you to do drop seek and read out what you've printed to that region and figure out where that cell was by the identity of the read strand. So we're going to call these zip code sequences that we bring in to add onto the surface of the cells. So the basic thing that this allows, this photo caging, is for us to do the following. If you're normally imaging and you're watching your tumor biology, you're usually sending in, say, a blue fluorescent epi-illuminated light source. And so this is the shown at the top right is the illuminator of a microscope that goes in and hits a dichroic mirror and goes into your sample. Your sample fluoresces green, and then it goes back through that dichroic and goes to a camera. So that's the simple light path of a microscope. Here, what we're doing is we're sending additional UV light off of a, if I were there physically, there would be an LCD projector that I can point to, and the LCD projector has a chip in it that is turning on and off pixels, which is why something like this would be illuminating onto the screen, is that those could be flickering on and off. And so you could put a mask like that. You can put the same exact chip that's in an LCD projector into a microscope. And in so doing, you can basically have an illumination mask that turns on light in only certain regions of the field of view of the sample. And so that allows us to do the following. We can take a plate of T cells that have, for example, these antibodies on it with the overhang. And if we illuminate the left-hand side and we add a blue zip code strand, we'll print blue to that. We'll print the blue zip code strand to those cells, and it'll be on their surface, and we can recover it and drop seek. Maybe later we come and we hit with red, a light source in the center and add a red zip code. And maybe later then we come and we illuminate the right-hand side and we can add a green zip code. And so we can essentially have 3 regions from 3 prints. And by the way, these little spurious cells, these are T cells that were set down onto a plate, and some of them float around. So that's why there's a little bit of spurious labeling. What's really cool about this is that we can go much faster now. And I won't show you too much of this today, but it's kind of the future of being able to do this very quickly, is that if you take these printables barcodes, and every time you use a print, so imagine this is ink, instead of having just the region that I showed you before, the print material, the ink, also has its own photo cage. Then you basically can start to repeat this process over and over and over again. And to see how that works, I'll show you down here again, a plate of cells. And if you illuminate on the left-hand side and you bring in a blue dye, then everything on the left-hand side of the image will be blue and everything on the right-hand side will be black. But now if you do stripes and you bring in a red zip code, you can see that obviously this leftmost region had blue before and now has red, so it's purple. It's blue only, this is red only, and this is black. And then you can do the same thing again, for example, illuminate just exactly half of the field and bring in a green barcode strand, and now you've created 8 different regions. So the formula for this is 2 to the n populations get labeled. And so what this basically means is that with 3 print steps, you've given yourself 8 barcodes. So if you do 8, you get 256, et cetera, et cetera. So you can really rapidly get every cell its own barcode. In fact, we can get to the point where we are pretty much at single cell resolution here. This is done, this is a 20 by 20 matrix that was laid down, and you can see just from 3 print steps, you can see green only cells, you can see blue only cells, you can see purple cells, etc. Okay, so just using that in the tumor microenvironment, just as a very quick example, if you take a tumor that has, for example, a peripheral region of T cell accumulation, they're marked with, cells are marked with GFP, and the center region of this is the red labeled tumor, you can apply barcodes to the outer region of this versus barcodes to the inner region of this, dissociate that tumor. And by the way, this is just for those that are interested, this is the process by which we do this. We inject tumors, then the green T cells come in 14 days later, and then 4 days later, we can pull this tumor out and image it. If you pull those cells out, at least on the basis, if you look by cell type identity, so you just UMAP everything, it's a little bit hard to see what's on the border and what's on the inside. It's a bit mixed. But when you start to pull cell types apart, like for example, if you just focus on the T cells and then ask a UMAP algorithm to cluster them, you really see how the red populations, those are the ones on the inside, cluster somewhat separately from the blues. And you can figure out what it is that makes them different. And one of the biggest things that makes them different are exhaustion and terminal differentiation scores. So if we apply a score to the cells that were in the center, it's an exhaustion score. You see that the cells that are in the center are higher for exhaustion scores, higher for terminal differentiation, and you can even do DEG analysis for that. And there you'll see, for example, that cells in the margin have a lot more TCF7, which is associated with kind of stemness as it compared to those that start to express more of these genes, ID2, for example, that are associated with this terminal differentiation. So that starts to bring the idea that within even just a single tumor, that spatially there is maybe perhaps in this case kind of a gradient of exhaustion. So T cells going from one fate to another fate or one position. And what's cool about that is that other cell types also show that same sort of spatial distribution or change in their differentiation. So if you gate on the monocytes and the macrophages within that same tumor labeling, again, you'll see this is now a U map of just the monocytes and the macrophages, and you can really see how the red dots, the red cells that were from the center of the tumor, end up segregating separate from the blue. And even though we're not using the barcodes here to do that, we're just labeling. We're using the barcode to isolate the cells or to show you the cells. The thing that drives that distinction is actually monocyte to macrophage transition. So to give you a flavor of that, the left-hand side of the U map really was high in Ly6C, and the right-hand side was really high in C1q. If you do a traditional monocle analysis, you'll see that all the cells in this U map can be laid out in what's basically the differentiation trajectory for monocytes to TAMs. And if you look then on the bottom right here, you see that the cells that, the distribution of the blue tag, meaning that you're on the outside of the tumor, is really high on monocytes, and the TAMs are really highly enriched in the center of the tumor. So as T cells are getting themselves exhausted moving inward, the macrophage monocytes are basically differentiating to macrophages. And so those kind of spatial relationships, I think, are how we're going to also understand archetypes, is that specific regions of the tumor may be programming cells to become the thing that the tumor wants them to become, whereas perhaps they start as more immature and pliable cell types when they are in other regions. So that brings up kind of a lot of interesting things to think about therapeutically. Okay. So the nice thing, just to bring this up, and I think many of you who think about spatial transcriptomics will recognize this, is that these sorts of approaches allow you to also ask questions about how gene expression is linked over space between cell types and within cell types. And so just to give you an example of this, this is a case of a lymph node where we gave 4 concentric rings, so rings of barcodes. So the cells that are out here are inside the lymph node, and here are on the outside of the lymph node. So just series of concentric rings. Think of donut holes within donuts. And so, for example, if you take a gene expression like KLF2, which is in our hands always more highly expressed within organs and less on the outside of organs, one can ask what other genes within T cells, for example, mirror that. So you can just ask for distributions that look like KLF2, and these are genes then that are coordinately regulated spatially with that same spatial profile. You can also look for the genes that are anti-correlated, that have almost the exact opposite profile, that rise with the same sort of slope, while KLF2 is going downwards as you go, from inner outer. So you can do those things, and you can pick them also for other genes. For example, CCR7 is another one that tends to go from inside to out, and it looks somewhat like KLF2, but its actual distribution is different. So, obviously you can do these sorts of tricks to understand what sorts of things are coordinately regulated in a given cell type. And then what's really neat is to start to think about how the gene expression in T cells are discoordinately regulated or coordinately regulated with genes that are spatially distributed in myeloid cells or tumor cells themselves. And I think that's where we're going to start to understand the molecular hooks that tie these various different archetypes and these cell types together. Okay, so as we're moving forward, one of the huge use of this is to start to understand, what's different about these regions. A lot of imaging that you'll see that we do and others people do has this black space. What is that black space? And what's special about the cells that are even labeled that makes them be the ones that are involved in maybe this very reactive archetype here versus this one here? So I think we're in a position now to start to do that, and that'll be exciting for the next few years. Okay. So I'm going to pause and I'm going to transition to COVID. I think COVID has represented a huge, well, it's not, it's hard to say it's a huge problem. I was going to say it's represented a huge area of entry for a number of us into these same sort of archetypal questions as they apply across other kinds of immunology. And so we and a group of others at UCSF, and I think there have been other consortiums like these across the country, while the vaccine were being made, took on another question. So the vaccine biology is obviously very well established and has been evolving as well. But you think about the goal of vaccine is to teach the immune system, what to expect. There's a bigger question to my mind, which is why is it that 5, 10, 20% of people have this very severe response and others don't? Because that's equally important as a scientific contribution to thinking about how we're going to deal with both ongoing COVID, but also pandemics in general. So what is it about certain people that their responses to viral infections are so much worse? And what is their immune system done in that case? So this gets into the archetypal definition. What is the archetypal definition of their immune system versus those of people that make a mild, moderate response and leave the clinic very quickly? So in our case, we surveyed blood. At least for this study, we also, and others, surveyed endotracheal aspirate, for example. We took all the cells from patients that were either COVID positive or COVID negative and severe and mild, and just had a sort of a UMAP-based clustering of those based on gene expression. And so, for example, within the neutrophil compartment, it was very clear that there is a population of cells that are easily defined as immune stimulated gene driven, so signature of these, that are really associated with COVID positive patients, but they're also really associated with mild COVID positive patients. So you can find that population as a whole and quantify it, but you can also start to generate an ISG score, a score for these. And you find that across, in fact, all the populations within these, mild patients have a pretty decent ISG score in all of their different subsets. Whereas severe patients really are typically very deficient in ISG gene expression. So the sort of conclusion from this and a bunch of other cell types we looked at, monocytes, platelets, is that mild moderate patients that go home in a day really have much higher expression of this gene module or this collection of genes. And this was systemic in the sense that if we looked at the frequency of populations that express the ISGs in neutrophils versus maybe in T cells, there was a really nice Pearson correlation, and moderate patients were out here, had a lot of the cell types, and severe patients were right here, as were sort of people that had bacterial COVID, that were COVID negative. And that's also shown here as a correlation plot for all the different ISG populations, one against the other. So there starts to be this kind of systemic feature that just starts to define severe disease versus moderate disease. And again, that overexpression of these genes is present in T cells and B cells, and also even in platelets. And I won't get into this data for the interest of time. So this idea that every cell type we were seeing in the blood had this really distinct skewing made us wonder about systemic sources of that. And when you're dealing with blood, your first thought is serum. Is everybody bathed in the same cytokines, for example? And so we, after literally staring at this for a couple months, we started saying, well, maybe this kind of weird, but maybe serum is skewing these ISG formations. And it could be that there's a solute in there. It could be antibodies. We first thought solutes. Because the alternative pathways we're looking at neutrophils, whereas S100A12 driven. So that's a response to, that's often thought of as something that can be involved in zinc and iron responses. So we saw a lot of patterns that made us think maybe there was something different about the actual composition of these patients' blood. But in any case, we started to ask about serum, and serum is really weird in SARS-2 because the people that are severe, if you look at their antibody responses to, for example, the full spike versus days in the clinic or days post you surveying them, they're always higher, actually. So they have a pretty decent antibody response to the spike, and then similar to the nucleus capsid, as compared to the mild moderates. So you might have thought that, okay, these patients are having a hard time getting an antibody response made, but in fact, it's not the case. Their levels of what looked like they should be neutralizing antibodies. We haven't verified that every single one of these is neutralizing, but they're definitely binding the full spike, and that you see this distribution. So this is, there's a lot of ways we can interpret this, but this was weird to us because we thought this didn't look like what you might have expected. And I'll just point out that total serum levels in these patients is, moderately within normal range. So it's not like they're making tons of antibodies overall. And in fact, also, if we look at their binding to C1q, which is a measure of their immune complexes, while there's a modest increase in mild, moderate, and severe, there actually isn't tons of extra immune complexes that you can detect in serum anyway. But there is this weird autoreactivity that's present, and so severe patient serum stain the other immune cells. So if we just take serum from mild, moderate, and versus severe patients, we would find some of this patterns of staining to ISG-induced monocytes in this case. So just saying the patient serum seems to bind with some frequency, but that pattern is all screwy. So that all the different patients here, they're binding to, for example, cell populations that were not interferon stimulated versus interferon, whether they're CD4s or CD8s. There's a pattern of high reactivity, but no single patient really looked like another one in terms of their distributions of these things. So to us, this didn't look like a single autoantibody, but just like this kind of pattern of autoantibodies. And we wondered whether this has a functional role. And so we did this, what was at the time really a weird assay. We took PBMCs from healthy people. We added interferon, and then we added patient serum to see what that would do. And then we waited 24 hours, and we did an induction of a particular ISG gene here, IFID-M3 was the example used. So without any serum, this is without any pre-interferon, this is after interferon, so everything shifts left on the IFID side. And if you use control serum, that happens too. So if you use mild moderate patient serum, they still shifted, so you got induction of this ISG gene. But in fact, it was much reduced, in fact, almost completely abrogated in many patients that were severe. So there was something in the severe patients that were reactive. And I'll point out that we had to go back and do this because this came out, our work was just being submitted where Casanova's paper came out in Science. And but only one of our patients also actually has antibodies to type 1 interferon itself that would explain this result. So that's this guy right here. And that's the data shown here for just staining to interferon-coated beads. So while while some of the explanation from this could lie in this particular patient that they have neutralizing antibodies to interferon, most of them are dulling their interferon response, but don't have that. And it turns out to still lie in the antibody fraction. So even though this is this 1050 where the patient looks like an interferon. The antibody, the person has made antibodies to interferon. And all these other ones, if we pre-absorbed the patient's serum with protein A and G. So in red is not pre-absorbed. So this is the blunting of this normal response. These are patients, this is serum that's from mild moderate patients mixed with interferon and then put onto PBMCs, and you get the shift. Doesn't matter whether you pre-absorb it or not to a great extent. In the case of these severe patients, you can see the blunted response that's restored back to normal levels when we took out their antibodies. So we just basically pre-absorbed IgG, basically. So somewhere in there, the antibodies were inhibiting the ISG response, but they weren't against interferons themselves. So then the question is like, what's the mechanism for this? How is serum antibody interfering with IFNAR signaling? And so we first thought it might be a complement effect, but we took the serum from the patients and we did some kind of classic cobra venom or heat treatment to try to kill off complement, and nothing, that didn't change anything. But when we blocked FC receptors, here we used all 16, 32, and 64 all together. Again, we got the restoration. So the pink is the restoration after treating with those, or putting in blocking antibodies to those. So for every single one except the patient that had the anti-interferon antibody itself. So everybody else restored. So this says that the blunting effect lied in the ability of antibodies to bind to FC receptors. And it turns out, and though people have sort of seen some cross functions for different FC receptors, that FC receptors alone, when engaged, will negate IFNAR signal. And so this is done, it's what we've done here, and I'll just summarize it over here on the right because I know I'm a little bit short on time, is essentially if you cross-link on a PBMC, on a monocyte of particular interest, you cross-link different FC receptors. It's only when you cross-link CD32 into the mix. So this is 16 plus 32, this is 64 plus 32, and this is just 32 on its own. You can always see that 32 blunts the response. So this is the blunting of interferon. This is again the signature gene, IFID-M3. You blunt the response to interferons when you engage 32. And it turns out that if you come back then with patients and you simply block their 32, again, you restore their IFNAR signaling. That's shown in a series of plots here. So for example, this is IFID-M3 expression in patients, and then these are the severe patients in the red that get inhibited for IFID-M3, and then this is that restoration when you block here, all of them, and here is just the same effect when you block just 32. So this brings up the idea that essentially in patients you have this somehow a pre-existing response perhaps, or one that is initiated in the context of severe viral infection that that directly negates interferon responsiveness. So I'm not going to go a lot further with that today, except to say that this starts to bring up really interesting questions reflecting back on the tumor archetypes. Is to start to think, we've really thought of antibodies as not being a positive effect on tumor microenvironments. But the story of having autoantibodies that subtly tune the archetype that you're making in a viral response starts to bring up the question of whether the same thing could be happening in some of our chronically cancer-ridden patients. So I think that brings up this question that I think will be really interesting to follow up on, and I want to use that as an area to conclude the talk and thank the people that have done all the work. So the COMET study that I just told you about was really one of these huge conglomerations of people that wanted to do something, and the lead authors are shown here, and this really couldn't have happened without a huge team that was largely led by Carolyn Caffey at UCSF, including a few others. And then the work that I told you about archetypes in general really has been focused in my lab. Kevin Berry did the work on the CC1 and K axis. Ken Hu did the ZipSeq that I told you about. Alexi Combs has been building these really large models of the dominant archetypes, the ones that are dialed in by the tumor microenvironment. And these folks started out the projects. We've had a huge array of collaborators, and I just mentioned some of the ones that I've specifically mentioned today. Really been helped by this UCSF-wide initiative to profile cancer patients. That's been a huge resource to be able to pay into, and we've been very fortunate with funding. So at that point, I'd like to thank you for your attention, and I'd be happy to talk both now. And I think one of the things that I want to emphasize about where we are with these virtual seminars is that whatever questions don't get answered today in the chat, please feel free to email. I can't be there, and we can't kind of hang out and drink a beer or drink coffee together. But to the extent that we all do want to kind of share science, please feel free to email. I'll respond as fast as I can, and happy to banter back and forth a bit with people that are there. So thank you very much.
Thank you. So Jess, I just want to emphasize regarding what just Dr. Cramo said, the questions that are already posted here and we don't have time to go over, they will also be sent to him. I think I don't want to finish without any questions, so let me try to read one or two. So Nelly Poliak is asking, CD14 has a polymorphism that correlates with severity of Kawasaki disease. Does the behavior of CD14 cells correlate with this polymorphism?
I don't know the answer to that question. I don't have any sort of, I don't think I have any data, and I don't think anybody else does, to particularly in the Kawasaki case. So I'm sorry, I can't answer that.
So Brian Miller is asking, do you agree with Ken Murphy's recent work showing DC1s are also critical for CD4 T cell activation? Does this data change your archetypal models?
Yeah, so this is one of these areas where we get into something that seems like a conflict and really isn't. If you take different mouse models of cancer, you will find that different cell types start to behave differently. I think Ken, so just for those on the call that don't know this work, so Ken recently published that sort of what seems to be in opposition to what Mikhail Binavi's published in our lab. So what we really saw was that when you eliminated CDC2s, you don't prime CD4s. And Ken sort of did a somewhat similar result where he said in a particular model that he was studying, if you eliminated CDC1s, you don't prime CD4s. In our hands, the CDC2s are dominantly the ones that also you can pull them out and they will stimulate CD4s in vitro. So I think this comes down to the idea that different cell types given a certain name may be somewhat different in different contexts. And so I don't disagree with his result. I think that the evidence in the humans says that there's a really high association with CDC2 function and CDC2 numbers and CD4, let's say the CD4-ness of the tumor response. But I think CDC1s are not all the same. And in different models, they will start to express different genes and do different things. We know that CDC1s are absolutely critical, and this is a story that we published in Cancer Cell this year, for a kind of an interesting function. They go to the lymph node, and then through this packet exchange, they distribute antigens to other cell types. So for sure, CDC1s become important for CD4 priming in some settings because they hand off the antigens to other dendritic cells that can then do the priming. So I think there's a lot of biology that goes on here, and I do think that the dominant story is CDC2s and CD4s. But just because there's a dominant story biology, this is like straight out of Jurassic Park, we'll find a way kind of thing. I think that there's all kinds of variations on these themes, and part of our goal as a community is to both see the dominant ones and then also see the dodges that the immune system can do in different settings. And I tend to think that what Ken saw was more of the dodge, but I guess that's also where these big profiling studies that we're all doing start to become very important to understand what's some of the dominant biology and what are some of the variations in patient world. You and I know when we go to the clinic, they almost take the dominant thing. You just have the flu, but knowing that there are variations in that look very similar but have deviation becomes important. And for the specific case, so it's kind of a long answer.
Thank you so much. So I think we ran out of time. I just want to thank Dr. Krummel again for this amazing presentation, and thank you all for attending the seminar today. Thank you.